My dear students, today's class is about the assimilation of food. Assimilation of simple food. Assimilation is the process by which simple organic food materials are absorbed and enter into our tissue and cell where they are involved in the cellular formation of cellular substances. Assimilation. What is assimilation? Assimilation is the process of formation of something new in the cell. Which materials are assimilated? The substance which are formed during and after digestion. You know that complex carbohydrate is converted into monosaccharide, that is glucose, fructose, galactose and so on, ribose. Similar way, the proteins converted into amino acid. There are 21 types of amino acids: lysine, leucine, isoleucine, methionine, threonine, valine, tryptophan are the most important essential amino acids in our body. Third is the fat or oil. Ultimately, at the end of the digestion process, fat and oil transformed into fatty acids and glycerol. These are three simple forms. Assimilation is the process when these three simple, diffusible and absorbable material enter into our blood and they go into our cell and tissue where they are involved in the some cellular materials formation. Monosaccharides generally they enter into our capillary, present in our villi, and through this villi, through the capillary in the villi, the capillaries in the villi, they enter into our directly in our blood system. Then they enter into our liver. Liver is the main organ where all the food materials, simple form, enter into the liver and they are utilized, they are stored, or they are involved in the formation of other substances. Monosaccharides, they enter into the liver and form the glycogen. This is the formation of glycogen. This process is called glycogenesis. Glycogenesis is the process by which glucose is converted into glycogen in the liver as well as in some cases it is the muscles. This is the glycogenesis. The most important organ is the liver. When the sugar level in our blood fall during excessive exercise, during running, what happened? In this case, the glycogen, sometimes, in most cases, they break into their substituent components. These are the glycogen from glucose. This is called glycogenolysis. This is glycogenesis. This is called glycogenolysis. These two processes are opposite to each other. Sometimes, in other cases, glycogen also be prepared in the liver in a process called gluconeogenesis. In some cases, when amino acids enter into the blood, it go to the liver, then it is converted into sugar, monosaccharide, glucose, then converted into glycogen. This process is called gluconeogenesis. Amino acids or fatty acids, that means organic substance other than monosaccharides, other than carbohydrates, converted into glucose. This process is called gluconeogenesis. Gluconeogenesis. These three processes generally continuously occur in our liver. And in which pathway the simplest substances enter into the liver? The pathway is called hepatic portal. Hepatic portal. Hepatic portal vein is a vein that originates in our viscera, in our digestive canal surface, and they carry all the simple digestive materials to the liver directly, where they anastomosis and ultimately they form hepatic vein. Hepatic portal vein originate from the intestinal different parts and they enter into the liver, and ultimately all the materials which are pass through this vein, they reach into the liver, and liver assimilate them and form different substances. The root is called the hepatic portal. This is called hepatic portal vein. Through the hepatic portal vein, the carbohydrate, monosaccharides and amino acids are transferred and reach into the liver for their final destiny, for their final function. Second point is the amino acid. Amino acid is called a compound having two functional groups. That is, the amino acid contains the A, C and R, CnH2, COOH. These are the main structures of amino acid. Though when the amino acid enter into our blood, they go to our liver, and when they enter into our liver, they have probability two functions. When the first function is the deamination, removal of amine group, deamination done by different enzymes, deaminase and transaminase. These enzymes present in the liver tissue transfer amino group to other component. In this case, the keto acid is produced. Keto acids is produced. This is one function. Sometimes in this amino group generally take part in the formation of the urea cycle. Urea cycle is the process. This is called, another term of the urea cycle is the ornithine cycle. Ornithine cycle occur in their in the liver tissue where excess ammonia that is formed during the degradation of amino acids are utilized to form urea in combination with arginine. Excessive ammonia in bind with ornithine forming arginine and amino acid. This arginine amino acid is broken into forming two components. One is urea and second is the ornithine. So the cycle is called urea cycle. Then the other point is the fat. This is the amino acid, fate of amino acid. First is the amino acid, amino group amino acid transferred into deamination process, and other remaining part present the keto acid. This keto part may be involved to formation of fatty acids, or it is utilized in the formation of sugar skeleton. This is the metabolism of amino acids. Third is the fatty acid and glycerols. You know, in the villi, the fatty acid and glycerol do not enter directly in the venous blood. How they incorporated into the venous blood? The process is here. It was described earlier that the fatty acid and glycerol enter into our lacteal of the villi. This is lacteal. Enter into the lacteal of the villi. How they are incorporated into lacteal of the villi? Initially, they incorporated into the epithelial cell of the villi, where amino fatty acid, glycerol, cholesterol, lecithin, as well as some apoprotein are aggregated to form a chylomicron. Then this chylomicron is delivered in the lymphatic duct. This is called lacteal. Through this lacteal, this lymph and the lymph substances, the food material, particularly fatty meals, they pass to the lymphatic duct and ultimately they drain into the thoracic duct. Thoracic vein. Through this thoracic vein, they ultimately reach into the venous blood system. So this is the fatty substances. What is the aim of the fat? What is the ultimate fate of the fatty acid? This fatty acid then go to the liver, where they enter into our fatty substance, deposit fatty substance, that is they are converted into triglyceride, or in some cases it may be converted into triglyceride, or it may be converted into cholesterol, or it may be converted into lecithin. In this way, the fatty substances are converted into complex form. During the formation, sometimes other complex organic fatty substances are produced. This is called the HDL, LDL, VLDL, that is the lipid associated with protein. They form complex biomolecules. These are very important for our fatty substance delivery in our venous blood supply. And sometimes these substances may form plaque in our artery and vein. And atherosclerosis in the heart vascular system, atherosclerosis if occur, then we are prone to heart attack. This is the deleterious effect of the excessive triglyceride, excessive LDL. These are the very deleterious and harmful substances produced in our liver system. So these are the sometimes when during starvation, when the excess fat are deposited in our adipose tissue or it may deposited in our liver. This fatty acid is oxidized, beta oxidation of fatty acids, and they form the acetyl CoA. And this acetyl CoA enter into the citric acid cycle and release energy. In this way, fatty acid may be involved for the production of energy during starvation. Although in normal case, glucose is the main source of energy, but during starvation, prolonged starvation and prolonged deprivation of the proper nutrient, the fatty acid degrade and calorie we get from this point. These are the more assimilation of food. This is the ultimate aim of the digestive system. If our digestive system do not perform their function properly, then these simple substances, some organic molecules will not be formed and our ultimate assimilation process will be hampered and we undergo a malnutrition stage. This is the main features. Later class I will discuss about liver.